Okay, so hello everyone, and welcome again to the 2026 ICPSR Data Fair at Love Data Week. We're happy to have you join us today for the webinar titled NACDA Study Highlight, the Vietnam Era Twin Study of Aging, VETSA. And now I'd like to give a warm welcome to our expert presenters, and we'll hear first from Dr. Sayan Kim, the director of NACDA. The floor is yours.
Hi, my name is Sayan Kim, and I'm Director of NACDA Program in Aging. And joining me is Catherine Lavender, the Data Project Manager for NACDA. We at NACDA help to guide data producers on data management and data sharing in the realm of aging populations. And I'd like to introduce Dr. Kerr Franz and Eric Buchholz for today's VETSA seminar. Dr. Franz, currently with the psychiatry department at UC San Diego, is an expert in the longitudinal study of stress and aging. She received her PhD in personality psychology from Boston University and completed her postdoctoral fellowship at Harvard University. Her extensive NIH-funded research focuses on risk and preventive factors for cognitive and brain aging. And Eric Buchholz is the senior research data analyst for VETSA at UC San Diego and a PhD candidate in information science at the University of Arkansas at Little Rock. He combines backgrounds in information science and health psychology to apply advanced analytic techniques to study health changes and risk factors over time. So together, Dr. Franz and Eric will provide an overview of longitudinal VETSA datasets. Then Catherine will discuss the use of VETSA datasets in NACDA portals. We will hear first from Dr. Kerr Franz.
Okay, so first of all, I'd like to thank Catherine and Sayun and everybody at NACDA. Just as you'll be hearing today, there's a huge amount of VETSA data, and they just have been marvelous in getting this processed and helping us think in ways of providing it to you in ways that will be the most useful for you. So thank you so much for that. So in 1999, two colleagues and I started talking about creating a longitudinal study, combining our expertise and interests in behavior genetics. That's Michael Lyons on the left-hand side of your screen at Boston University, neuropsychology, that's Bill Kremen, now at UC San Diego, and adult development and aging, that's me in the middle. After 3 attempts, NIA finally funded us in 2001, and the Vietnam era twin study of aging, or VETSA as we call it, was born. This was despite the reviewers' doubt that we would find much mild cognitive impairment or aging in people who were in their 50s at our baseline assessment. But 25 years later, VETSA is still going strong, and we did find mild cognitive impairment in people in their 50s. I used to think of Michael, Bill, and I as like the Mighty Morphin Power Rangers. I don't know if you ever watched those shows. I did, but by our powers combined, that with our unique set of interests that we were able to make VETSA happen, along with, of course, the support of NIH and NIA. So next slide, please. So what is VETSA? It's a longitudinal study of early identification of risk and protective factors associated with cognitive and brain aging. Our original focus was on the behavior genetics of normative aging. The study was largely modeled after the seminal work on cognitive aging of K. Warner Shai and the Seattle longitudinal study, with really in-depth looks at cognition, but also a thorough assessment of all the different aging factors, risk factors that might be possible. And the different slant that we had on cognitive aging was really twofold. One part was we were looking at behavior genetics. Is cognition, cognitive change, heritable, genetically influenced by how much? What is modifiable, what is not modifiable. And I'd also like to give a shout out to my mentors in adult development and aging, who include Abby Stewart and David McClelland. So ultimately, now we have collected 4 waves of data collection with a baseline at midlife. VETSA 1, the average age was 56 years with a range from 51 to 60, and up to VETSA 4, in which the data collection was just complete when they were on average 74 years old with a range from 66 to 78. And as I'll be telling you later, the VETSA 4 data collection is complete, but it's not yet at NACDA. Next slide, please. So what makes VETSA unique? So first of all, we have this narrow age range. The sample is only 9 to 10 years in age range. And this gives a greater ability to examine within individual change over time, and within individual and between individual change. It also means that we and you can conduct both wave-based and age-based analyses. Second, even though you only get one NIH grant at a time, it was a planned longitudinal study starting right at midlife. This was a nationwide community dwelling sample. They were all randomly recruited from the same cohort at the Vet Registry Archive. And what is the Vet Registry Archive? So this is the Vietnam era twin registry archive, which is now in Seattle, Washington. And what they had done was they had identified all of the twins who were in the military between 1965 and 1975. Doesn't mean that they went to war, but this was just called the Vietnam era. And they had over 10,000 individuals in their registry. And that's where we randomly recruited this sample from. Because we were dreaming of doing a longitudinal study, following the wisdom of people like Warner Shai, we made sure there were consistent methods and measures in every wave, and that these would be strong even as people were aging. So, we were trying to keep in mind what people might be able to do 20 years after they started. In addition, we collected what we call attrition replacement subjects at waves 2 and 3. Why did we do this? Anybody who's taken an SAT or a GRE, you practice, you do better with practice. So the more times you take a cognitive test, the better you're likely to do. And so we wanted to be able to assess this because really very few people had looked at it in aging samples. They look at it in young people, but not in older people. And so we added extra participants at waves 2 and 3 so we could be able to do this checking of practice effects in cognitive aging, but also it helped us keep the sample size large. And finally, what makes it unique is that it's a large all-male sample, and we can talk some about this at the end. For us, the purpose of this was that it was adequate power for twin analyses. You need twice as many people with twin analyses because you need both twins, MZs and DZs. And why is it male? Partly because most of the people who were in the military during the Vietnam War were men, and there were just too few female twins to have a large enough sample. Next slide, please. Very simply, the VETSA sample breakdown at baseline, we started with around 1,237 subjects, 82% retention at VETSA 2. As I said, average age 56 at baseline. The total individuals with the attrition replacement was 1,608. They very much were representative of men in their age group in the United States, average education of around 13.8. 78% were married, 88% non-Hispanic white, and the majority, about 80%, never served in combat. Next slide, please. And just so you know, about 20% of all men age 65 and older served in the military. So, they really are part of our population. What is our basic method? So participants traveled to either UCSD or Boston University. They stayed 2 to 3 nights on site, depending on which leg of the project they were in. 1 month before they came, we mailed them this 55-page psychosocial questionnaire, which we told them to bring back in with us. So it means we have really like 99.9% completion for the psychosocial questionnaire. On the day of testing, they came in at 8 o'clock, got consented, had a blood draw, sat down for breakfast, and then they spent the rest of the day with us doing medical history interviewing, in-depth cognitive testing, and physical functioning measures. The next day, if they were eligible, they would have an MRI brain scan. And not everybody was eligible for that, so that's why they stayed the extra day. On your right, there used to be a flow chart of the data collection and attrition, and I put this here to scare you because you're always less scared the second time you see something. So this looked like a massive octopus, and you will find it on the NACDA site. But what this is our flow chart. And so if you go in and look at this, don't be afraid. It looks hideous, arrows all over the place, but what it shows to you is the flow from VETSA 1 to VETSA 2 to VETSA 3 with the attrition and the reasons for attrition and the attrition replacement, the MRI sampling, that kind of thing. So don't fear. It's just a flow chart. Next slide, please. There are other data, VETSA data available, but they're not yet at NACDA. So the VETSA 4 aging data, Eric's working on to get it to you this year. There is magnetic resonance brain imaging, the MRI data at all time points. And this will be processed data because I think NACDA doesn't have space for actual brain scans. But that is in the process of getting it into NACDA. There is air pollution data from a grant that we just finished working with people at USC that gives fine particulate matter, 2.5, NO2, and ozone for the bulk of the subjects who had participated in VETSA 1 and VETSA 2 from 1993 to 2017. Why those dates? Because that was the scope of that grant for that period. And we also have blood-based biomarkers. So for people who are interested in things like inflammation and AD biomarkers, we were able to get those sorts of biomarkers processed in VETSA 3 and VETSA 4. So those will come. And there is a link available to take you to the VETSA UC San Diego project website where you can see about how to contact us if that's something that you want now. Next slide. There is also other participant data available, and this is from the VET Registry Archive. So if you're interested in a true life course perspective, not just aging after 50, but we're aging from the minute we're conceived, if you think of it that way. We don't quite go that far back, but the Vietnam era twin registry was doing surveys and things on its own, not NIH funded for the most part. So there is archived survey data from age 20, age 38, age 40, age 42. They are the owners for privacy issues of the genetic data. So GWAS from all of these men, zygosity, and they also have stored blood specimens. And there is a link to them to find out how to access that data from them. So there's way more to explore even beyond the data that we're providing to you. Next slide, please. VETSA takeaways. This is a high quality longitudinal data set with a large number of the same phenotypes at multiple time points. Although our focus was on cognition, there's plenty there. Plenty of phenotypes important for aging and human development, on health, lifestyle, personality, exposome, cognition. The study was NIH funded for over 25 years now. It's a proven success. We've published over 250 journal articles. We're a member of national and international consortia. So these are strong data with a history of strong publications to help you with your dissertations and whatever work you're doing. There's no need to worry about knowing how to do genetic or twin analyses. The majority, probably 90% of VETSA papers are not twin papers. We look at individuals. There, you simply adjust for the effective family in your statistical models. But if you want to do genetics, you can obtain the genetic data through the vet registry. And finally, we're keeping on going, not by me, but our new generation of PIs. VETSA 5 has been submitted. So next slide, please. So now Eric will talk about the VETSA data.
All right. Thank you very much, Carol. All right. Next slide, please. All right. So how do you find your way? Well, my goal is to help you find your data because the forest is dense. And I want to go after two questions. One is what will you find in the data files? And two is with your research question, where do you go? Next slide, please. So for the first question, what will you find in the NACDA data files? There's two types of data files. One is the raw data for each wave, and two are the data files that condense some data across all three waves for all participants. Next slide, please. And for the first type, the raw data for each wave, it's at the item level and the composite level. So if you looked at depression, you could get separate items, or you could get composite scores, which are subscales. And this includes the psychosocial questionnaire, or PSQ, medical history, and the data entry sheet. And I'll go through each of these in turn. Next slide, please. And so the psychosocial questionnaire covers a lot of different topics that range from health and childhood family background to work, social networks, intimate relationships, and various leisure activities like sports, books, and walking. Next slide. Next is the medical history interview. And so this includes a variety of things like hearing, sleep, tobacco use, alcohol use, head injury, abdominal pain, urination, and sexual functioning, as well as medications, health problems, weight change, breathing problems, hospitalization, heart issues, and joint pain. Next slide. And then next is the data entry sheet. And so this includes all raw data from the day of testing. So this is raw cognitive data. There's also physio measures like the audiometer, blood pressure, and height and weight. There's physical functioning like the walk test, rise from chair, and grip strength. And then we also collect blood chemistries like the basic metabolic panel, complete blood count, inflammation markers, and lipids. Next slide. And so for the other type of data, this is data files that condense some data across all three waves for all participants. And so this involves the administrative file, the AFQT, or the Armed Forces Qualification Test, which measures general cognitive ability and process cognitive data. Next slide. So for the administrative file, you can get how old people are, education, ethnicity, race, occupation, year of death, APOE status, when they're born, their handedness, and also we have different types of wave coding. Next slide. For the Armed Forces Qualification Test, again, this is a test of general cognitive ability. It's given at all three time points, and individual items or scoring is not permitted by the Pentagon. So instead, we are providing overall scores instead. Next slide. And the process cognitive scores are a variety. This includes the mental rotation, trails, matrix reasoning, CVLT, AFQT scores, digit span, and spatial span, as well as letter number sequencing, logical memory, visual reproduction, hidden figures, Stroop, verbal fluency, reaction time, and the AX continuous performance test. Next slide. So that is a lot to take in. So let's return to the second question I wanted to ask, which is, with your research question, where do you go to find information? Well, there's some documentation. This includes the actual questionnaires, the README files, and the operating manual. And there's also a handy-dandy crosswalk file. Next slide. So what is in this crosswalk file? So there's three tabs at the bottom that help organize things, the PSQ, the medical history, and the data entry sheet. And then on each of these sheets, you can actually see the question and the variable names for each of the waves. So wave 1, wave 2, and it keeps going on to wave 3. Next slide. So some things to consider. Not all variables are used at all time points. In addition to the crosswalk file, you can look at a list of differences for each data set to see those changes. For example, if you look in the documentation PSQ differences file, you're going to see some different tests. So if you look at the well-being test, it's present at wave 1, 2, and 3. If you look at resilience, just wave 1. And if you look at ways of coping, it's just wave 1. Next slide. Some important variables to consider are the CID, which is the participant ID, And so this is composed of the first 5 digits, which is family, and then the 6th digit is the individual. So it's a 1 or a 2. So CID case is the family portion of that. CID twin is the last digit, the 1 or 2. So if you had 2 brothers and you have like a CID of 988931, you get the CID case, which is 98893 for the family, and then the twin, which is going to be 1 or 2. Next slide. So VETSA is a really cool project I've been including in my own research, and I'm interested in close relationships, health behaviors, and health outcomes. So I want to do something with it. I look at attachment, how emotional bonds shape how people relate, regulate emotions, and seek safety in relationships. And so my research question is, if attachment predicts health outcomes and cognitive decline, how? Through what pathways? Next slide. So there's a lot going on in this model, and this is looking at different variables across time. We're taking advantage of the longitudinal nature of VETSA, looking at age 56, 62, and 68. So we looked at how attachment at age 56 predicts different variables at age 62. These include perceived stress, doctor visits, smoking, drinking, and social isolation. And then these in turn predict frailty and mild cognitive impairment at age 68. The basic takeaway message is that perceived stress is an important mediator between attachment and frailty and mild cognitive impairment, likely through wear and tear in the body. And this is being published in health psychology. Next slide. That's just a description of what I described for the model. So you can go on to the next slide. And so overall, can you find your way through the data? Well, make use of all those resources. Look at the READMEs, the crosswalk, the operating manual. And again, as a reminder, there are 2 types of data files. There's the raw data for each wave, and there's the data files that condense some data across all 3 waves into a few files. Next slide. So I want to thank the VETSA twins, the VETSA investigators, staff, and NIA. Next slide. And I will pass this on to you, Catherine.
Thank you. Thanks, Eric. That was really interesting. I'm going to talk about accessing the VETSA data using the NACDA site, and I am Catherine Lavender, the NACDA Data Project Manager. And so I wanted to tell you a little bit about NACDA first. We're the National Archive of Computerized Data on Aging, and we've been around for more than 45 years. And so we've been preserving social science data and sharing it all that time. And we're part of ICPSR, the Inter-University Consortium for Political and Social Research. If you're here today, you probably already knew what that was, but just in case you didn't, we are funded by NIA. And because we're funded by the federal government, you do not need to be a part of an ICPSR member institution to access data from our site. We do host mostly public use quantitative data available at no cost. But just because it's free doesn't mean you don't need to do anything. You will need to register for a researcher passport account in order to download data files. And I'll talk a little bit about that. So to get to the VETSA data from the NACDA website, you would visit nacda-aging.org and use the search function from the homepage and type in VETSA. And the results will lead you to the VETSA study homepage. The link to the study homepage will be the full title, the Vietnam Era Twin Study of Aging, VETSA, United States, 2002 to 2019, and that study number is ICPSR 38836. This study page is where you're going to find out everything you need to know about the data before you download it. And the default view of the study page is going to be what we call the At a Glance tab. And this includes descriptions of the methodology, sample, and more. So from that study page and the At a Glance tab, you'll see that there are other tabs, and you're going to want to review these other study page tabs. There's a variables tab, and you can see a variable listing and some of the basic descriptions of the variables and even some frequencies. And you can learn about what other people have published using the VETSA data by clicking on the data related publications tab. But when you're ready to download, what you want to do is either use the download button to the top right of the study page and download all of the data files available for the study in a specific format. It'll give you the options for like SPSS or SAS. Or you could use the data and documentation tab, and that's where you can choose which data set you might want, or just other specific individual files. So the data and documentation tab, you'll see that it's organized to match the VETSA team's intentions with those files that are merged across waves and files that are unique to each wave. And the names of the data sets match the outline that Eric described with the administrative data, the AFQT data, and so forth. And you'll see, from this tab, their size. And so, for instance, the study level files, there's 14 megabytes there, and there's an option to preview or download the files. And the same is true for the rest of the files listed in the data and documentation tab. And you can preview the documentation here and select which items you want to download, again, individually. So to download data, though, you can look at documentation, and I think you can even download some documentation before being logged in. But to download data, you do need to be registered and logged in. And as I noted, you can review variables from that variable tabs without being logged in. But once you decide you're going to download data, you'll go to your ICPSR account and either create an account or log in using Google, your organization credentials, OrgID, or LinkedIn. And then what will happen is you select the files you want to download, and it'll prompt you to agree to our terms of use. And the terms of use are just a statement that you will not try to identify respondents, you will not redistribute the data, and that you will properly cite the data. And once you get a hold of these slides, you'll have links to all the resources, including how to create an ICPSR Researcher Passport account from the ICPSR site. When you download any materials, you'll get a zipped package. And so you accept the terms of use and your download will begin, and you'll find that in the folder you selected or in your downloads folder, you'll see this zipped package, and it'll be named ICPSR plus the study number and the version .zip. So in VETSA's case, it will be ICPSR 38836-v1.zip. And these zipped packages, we always get questions about, why is it a zipped package? And we do this because it allows us to include important metadata and documentation with your download. The download, once it's unzipped, what you'll see is all of the files and study content that you selected, and that's going to be within the zip package. And the data files will be located within dataset folders named like DS0001. There will also be study level documentation included at the same level in the zipped package unnested. And this is where you would find the crosswalk that Eric mentioned and the flow chart in the documentation that Carol mentioned. And so it's included at this level, not nested within the data set folders, because it applies to all of the data sets, not just to a single data set within the study. So once you've downloaded and used the VETSA data in your own research, it's really easy to cite. You would just go to the study homepage, and again, it defaults to that at a glance tab. So you're already there. Scroll down to the citation section, and you can copy the citation directly from this page, or you can export it. There's hyperlinks where you can access like RIS or EndNote formats. So, also, if you've already used VETSA and you're published and you want to send us your citation, you can send that to icpsrbibliography@umich.edu, and just make sure to include the titles of the studies you use, in this case, VETSA, along with the study number or study DOI, and provide a link to the full text or attach it to an email, your publication citation, that is. Okay, So, if you want to stay in touch, uh, if you really enjoyed today and you want to know more about NACDA, we have a QR code here and a short URL. It's https://myumi.ch/3RDDP. Um, Again, all of the links will be provided in the slides and we will, send those out to people who registered. But this QR code, this short URL will get you quick access to the NACDA site, to our Collectica portal, and you can find out more about that on our site, to our YouTube playlist, our newsletter and sign up, and some more NIH data sharing resources and our social media. If you'd like to learn more about VETSA and VETSA-related research, you can look up in PubMed, Franz and Kremen. And there's also, you can search the ICPSR site, the VETSA website, the VET registry site, or you can just email us at icpsr-help@umich.edu or email VETSA at vetsa-data@health.ucsd.edu. And so now I am going to change the layout again so that you can see us, and we will go over some questions. And thank you so much, everyone, for being here. I'm going to go ahead and read the questions and, to our presenters, please jump in as you need to. And so our first question is any of the data restricted use?
So I think there are different definitions of restricted use. Right now, none of the data that we've submitted to NACDA is restricted use and by their definition. The vet registry, as any data archive, does have rules about who owns what data. And so because of the preciousness, and these are veterans, there are VA rules to cover, there are data that are only available through them. So there's partly that, and that list of vet registry data I gave you. Some of the data that are at the vet registry are not restricted per se, but they weren't collected with NIH funds. So they're not required to make them public as, newer NIH grants say. So the vet registry views itself as owning the genetic data, the raw genetic data for the research participants. Some other things simply aren't on the website yet, like and probably never will be, because like the MRI scans, which would be more than 2,000 of them across time, the raw data would simply, I think, overwhelm, my understanding, NACDA capacity. And so if you actually want the raw data, just write to us and we can send that to you. Instead, what we're working on providing to NACDA are, highly kind of professionally processed with all sorts of information, scans so that you can get, volume, whatever kinds of dynamics you want, usually based on free surfer. So that's something that's in process. So, and it's my understanding probably NACDA will never be allowed to have the raw genetic data. so I don't know what policies are about that. So
Thank you, Carol. Yes, like I noted, we host mostly quantitative public use data. And, for years, ICPSR has been a quantitative social science data archive. But every now and then, we do try to see what other types of data we can host. And we do have some qualitative data and some other data types where there have been, specially sponsored projects to help us expand our capabilities. And who knows what will be available in the future if we are ever, you know, um, more capable of making available image type data or other things, we'll let you know. But I think that for what we offer and what you've described, that people have the access that they need.
Yeah.
It's just, kind of navigating these different places and determining what is going to be most beneficial for someone's research. We do have a couple more questions. So I'll go ahead and this one is, can I still use the data if I don't know how to do twin analyses?
So I touched on this before, and the short answer is yes. That quite honestly, the vast majority of our papers are non-twin papers, and they're just simple statistical methods that you can use that basically is just going to kind of make the p-value stiffer for you in order for you to make that correction for the nestedness within family. And so don't let that stop you. If you want to do twin analyses but don't know how, there are wonderful resources at Virginia Commonwealth University and other institutions out there. And then you'd have to work with the vet registry to get the some of the data that you would need to be able to do those twin analyses. So you don't need to do anything fancy, just run your regular regressions and whatever, and just correct for family. And there are other studies I know at NACDA where you have family members, and you just have to adjust for that.
Yeah.
Anything on that, Eric? Like your paper isn't a twin paper.
Right. We don't control for that. We don't use random effects, but yeah, other papers I've worked with random effects on. But you can get by with regressums in general.
Thank you both. I think we'll take one more question. And Eric, you talked a little bit about this, but in the organization of the materials available, but somebody asked, why isn't there just one big data set? Like some studies hosted at NACDA are just a large longitudinal data file that gets updated each time a new wave is added. So what made you all decide to create these individual pieces, these different data sets that comprise the study?
Should I answer that, Eric?
I wasn't sure which of us should answer.
Since it's probably my fault. So a lot of studies of aging have been survey studies. We have 10 to 12 hours of data on these research subjects at each wave. And I think we are literally talking about thousands, more than 10,000 variables, maybe, Eric.
There's a
And
lot.
then there's the raw level data, item level, for both like the personality measures as well as for the cognitive measures, and then the created scores. We have the addition of the attrition replacement subjects at the different times. So it's why we just left the organization for you in terms of you can download the data sets, pull out the variables that you want, link it with the admin data set so you know exactly, who entered the study when, and go from there. We felt it actually, and from our use, this is, like 20 years of our use, that we just felt that it was easier to not try to make sense of 10,000 plus variables.
Thank you so much. Yes, that makes sense.
You can create one big data set and tell us what you think of it.
Thank you for that. Yes, I think that makes a lot of sense. And Eric, sorry, I didn't mean to call you out that way. I was just you were talking about the breakdown. So but yeah, Carol, thank you very much. And
it was my fault, so
It's no one's fault. And I think it really depends on the research questions. And Noah, do we rarely need all of the variables, and it's thematically organized that you can include demographic variables and then cognition and then health related behavior variables for your research questions. So you can create your custom data set in a way.
Yeah. Yeah. I mean, I think you thought ahead about convenience of use, which is important. So already when you were creating the data sets, you were thinking about sharing it. So thank you so much for that. And thank you both for being here today. And I guess now we're at the point where we've reached the end of our time together. And I'd like to thank everyone for joining us, and to our presenters, and to our, the NACDA director, Sayan Kim, for the lovely introduction. And just thank you so much to everyone for being here, and enjoy the rest of your week.